The Linea IVT platform is a combination of two next-generation technologies that enable the production of better RNA faster. Enzymatically produced linear DNA in vitro transcription templates are a superior alternative to traditional plasmid DNA-based IVT templates. Produced through a completely cell-free enzymatic manufacturing process utilizing a single enzyme, linear DNA IVT templates offer rapid turnaround times and high purity without the additional requirements of plasmid DNA, such as enzymatic linearization and complex filtration. Unlike plasmid DNA, linear DNA IVT templates can amplify complex and unstable DNA sequences, such as poly-A homopolymers with high fidelity. Linear DNA is a blunt-ended dsDNA construct that is perfectly suited for IVT and can easily be chemically modified to meet specific needs. Modifications to linear DNA IVT templates permit use of a proprietary next-generation fusion RNA polymerase, which consists of a DNA binding domain and a high-fidelity RNA polymerase. The unique combination of linear DNA IVT templates with the fusion RNA polymerase offers significant advantages over the traditional pDNA-based IVT systems, including the prevention of dsRNA formation, higher target RNA yields, simpler, faster, and more cost-effective workflows.